Okay, hello everyone. I'm sorry I am absent again today. I had planned on coming back, but I just do not feel well enough to to come back to class today. So instead of spending an entire day doing book work, I decided that we are going to continue on with the notes. So hopefully this recording works. Today's lecture is going to be on those notes that I gave you guys on Monday of this week. It should be the green set of papers. If you need a copy, you can get them from the substitute teacher. I should have left some out for you guys. I don't have a copy for everybody, so hopefully most of you guys have kept your notes in your binder. Okay, so let's start. RNA is ribonucleic acid. Okay, notice that it is RNA. Okay, ribonucleic acid. It's similar to DNA, but there are three things that make it completely different from the DNA molecule itself. Okay, structure-wise, that is. Okay, so a ribonucleic acid, or RNA, has a 5-carbon sugar that's called ribose. Remember that in DNA, you have a deoxyribose. RNA also has a phosphate group and a nitrogenous base. So these two parts, the phosphate group and the nitrogenous base, are the same. The main difference is that it has a ribose group instead of the deoxyribose, like in DNA. RNA is single-stranded. That means it's only made of one strand instead of two, so it's not a double helix at all. And in RNA, instead of having thymine, you're going to have a nitrogenous base called uracil. Uracil. It's abbreviated with the letter U. Okay. And we'll talk about how it pairs up even though RNA is a single-stranded molecule. Now, when you have RNA, it is just a copy of a single gene. Every gene makes copies of just one type of protein. Right? So RNA, when you make RNA, it's going to be a copy of one of the genes in your DNA molecule. And that is because if you have RNA as a disposable copy, you can make thousands of RNA molecules from just one sequence of DNA. So if you have just that one gene on the DNA, instead of taking that DNA and using it and only making one protein at a time, you can essentially make thousands of copies of it and at the same time make thousands of proteins from that one DNA. Okay, so that's a benefit of it. Now, I know I'm going to be going a little fast on this, and if this video works well, then I will be posting it on Edline for those of you who decide, or who either lost their notes or are deciding to write all of the words down instead of using my skeleton. Okay, so this will probably be on Edline. Okay, so the types of RNA. RNA is going to be used in protein synthesis, like I've talked about. Okay, it's a copy of one gene from DNA. Okay, it's used in protein synthesis, and there are three types that are used in there. There's messenger RNA, or mRNA, and that carries information from the DNA to the rest of the cell. So mRNA is going to be like the boss. Okay, he gives the message to everyone else on how to assemble or how to put those proteins together. Okay, because synthesis, remember, means to make, right? So protein synthesis means to make protein. The second type of RNA that we're going to talk about is rRNA, and it is going to be found as part of a ribosome. Okay, remember we talked about ribosomes being part protein and part RNA, right? The RNA that's there is called ribosomal RNA, so it's pretty easy to remember. Ribosomes, ribosomal. It's found in the ribosomes where the proteins are going to be put together. And the last type of RNA is called transfer RNA or tRNA. tRNA is really important because it gets the message from the mRNA, the messenger RNA, and it's going to take that message and figure out which amino acids, which amino acids are going to be needed to make the protein that's coded for in your DNA, in your gene, essentially. So again, RNA, single-stranded, it's used in protein synthesis. There are 3 types. There's messenger RNA that gets information from the DNA. There's ribosomal RNA, which is going to be a part of your ribosome, and it helps put proteins together. And lastly, there's transfer RNA or tRNA. And tRNA is going to determine which amino acids to be used for the proteins. And it figures that out by getting the information from the messenger RNA. And lastly, remember that proteins are made of amino acids. This is why amino acids are so important. Proteins are just really, really long polypeptide chains. Hopefully you guys remember some of that from chapter 2. Poly means many. Peptides, amino acids, many amino acids all strung together in one large protein molecule. Okay. The process of making RNA is called transcription. Okay, to transcribe something, scribe means to write. Okay, so transcription, you are essentially taking DNA and rewriting it into an RNA molecule. And usually when we're talking about transcription, we're talking about making mRNA, but the other RNA molecules are made pretty much in the same way. Okay, so again, to go from the gene that's on the DNA to mRNA, the cell undergoes a process that's called transcription, or to rewrite the code, to rewrite that DNA into an RNA molecule. Sorry. Oops, I was supposed to press pause. Oh well. Okay, it requires an enzyme known as RNA polymerase. Just like how in DNA synthesis, when we make copies of the DNA, we're going to need DNA polymerase. In RNA synthesis, in the making of RNA or transcription, we are going to need RNA polymerase to do this. RNA polymerase is going to attach to the DNA at a region that's called the promoter region. Promoter, okay? It's the beginning of a gene, and if there is no promoter region, the DNA polymerase cannot bind to it. Again, the promoters are at the start of the gene, and it'll tell where the RNA polymerase should attach to it, and that is where the RNA polymerase is going to start its transcription process, or start making RNA. The first thing it does after it binds to the DNA is that it separates the 2 strands. Okay, in DNA synthesis, the helicase will do this, but in RNA synthesis, in transcription, where's my pointer? Transcription, okay, we are going to have RNA polymerase doing both jobs. It's going to separate the 2 strands of DNA. It's going to use one of those strands as a template to make the mRNA. And then when it reaches an area called the terminator sequence, it's going to stop transcription. Okay, so it starts in the promoter region when RNA polymerase binds to it, and then it's going to separate the 2 strands, use one DNA as a template, and then stop when it reaches a terminator sequence. Okay. So let's take a look at this. Here is your cell. Here is the nucleus with the DNA and everything. Okay, transcription begins when RNA polymerase binds to a promoter region. See this region right here at the very beginning of the DNA molecule is differently colored to represent a promoter region. Promoter region just has a special DNA sequence that's recognized by RNA polymerase. As the RNA polymerase binds, it's going to start unwinding that DNA molecule and separating the two strands. Okay, as it does that, it also starts to make a copy of that gene, whatever gene it is on the DNA, onto a single strand of RNA molecule. When the polymerase reaches the terminator sequence at the end of the gene, it's going to release the mRNA, and it's going to pop off of your DNA strand because it's finished. Okay. Here in the corner, it shows what I mean when I say thymine is replaced by uracil. If you guys look closely at this, you're going to see that if the DNA molecule reads T, T, A, T, G, G, okay, again, that reads T, T, A, T, G, G, your RNA molecule is going to read A, U, A, C, C. So instead of having a T to pair up with that A in the DNA molecule, it's going to have a U for uracil. Okay, so that's what I mean when I say that thymine is replaced by uracil. Again, RNA has 4 nitrogenous bases. 3 of them are going to be the same: adenine, cytosine, and guanine. The 4th one is going to be uracil. So instead of having thymine, your RNA molecule is going to be using uracil. Okay, again, adenine, instead of pairing with thymine, is going to pair with uracil. Oh, yeah, there's a pause button. So, for example, in this new example, remember that DNA is double-stranded. So when you have transcription and RNA polymerase starts to unravel the double-stranded DNA, it's going to only use one strand as a template. Okay, it doesn't need both strands because RNA is single-stranded. It means it only makes one strand. So let's try this, okay, in class. You guys give this a shot. I'll give you guys a few seconds to fill it out. If the DNA strand reads A, T, G, C, C, A, G, T, C, what do you guys think that RNA strand is going to read? Okay, I'll give you guys a few minutes, or well, not a few minutes, a few seconds to fill that out. Remember the rules. You still have these 3 bases, but again, thymine will be replaced by uracil. Okay, 30 seconds. Okay, did everyone give it a shot? Hopefully you did. Hopefully you got it right, and hopefully you just weren't sitting there staring blankly at the screen waiting for me to go on to the next one. Okay, so if your DNA strand reads ATGCCAGTC, your RNA strand is going to read UACGG UCAG. Okay? As I mentioned before, if it has adenine in your DNA strand, as we make the RNA strand, instead of pairing that adenine with a thymine, you're going to pair it with uracil. Okay, since RNA still has adenine in it, when you have thymine in the DNA strand, you can still pair it with adenine. It's the other way around that you can't have it with thymine. Hopefully that makes sense. Okay, so adenine is going to pair with uracil, thymine still pairs with adenine, guanine with cytosine, okay, etc., etc., etc. So again, U, A, C, G, G, U, C, A, G. Okay. Notice again the U replaces the T only in the RNA strand. Okay. Now, we're not done with making RNA. So far what we have is that the RNA polymerase comes in, it unwinds the DNA, starting at the promoter region, and it starts to make a single-stranded RNA by using half of that DNA molecule as a template. It ends when it hits the terminator region. Right? And just to remind you guys again, every time you have adenine in the DNA molecule, you're going to have uracil in the RNA molecule. Before you finish with transcription, the mRNA molecule has to be edited. There you go. Edited. Okay, so it has to be fixed before it becomes the final version of mRNA. So, after it's made, you have two different things inside of that RNA molecule. It has to get cut and then put back together. It's really weird. Okay, so there are regions called introns that are not going to be involved in any kind of coding at all. It doesn't do anything. It has to be cut out of the mRNA before the mRNA is going to be ready to be used for protein synthesis. And then the RNA, the immature RNA, also has regions called exons. Okay, they are actually involved for coding in the protein. So after you cut out the introns, there you go. After you cut out the introns, you're going to splice together the remaining parts, which are the exons, so that you have your final mRNA molecule. Okay. I know it sounds a little confusing, but hopefully the next picture will help to clear it up a little bit more. Okay, so here is the sequence. This picture here represents the immature RNA, this is the DNA strand. The DNA strand is going to have introns that are represented by the blue squares in between each exon that's represented by these brownish orange squares. Okay, so when you make a copy of the RNA, it's going to look exactly like this. It's going to have an intron, then an exon, then an intron, then an exon, then an intron, then an exon, then an intron, an exon, and so on and so forth. Okay, these blue regions that are introns need to be cut out of the RNA molecule. Oh, there's my laser pointer. Needs to be cut out of the RNA molecule before you can have a final mRNA molecule down here. Okay, so DNA, again, is going to have introns that are represented by blue in the middle of exon regions that are indicated by these brownish yellows. Orange, I don't know what color that is. When you cut out those blue introns, you need to splice together the exons. You need to put them together in order to have a full mRNA molecule. Okay, if it helps you to look at this picture, please feel free to color them in differently just so that it's easier for you to remember. Okay? I know in black and white it's kind of hard to tell. You know, scientists don't really know why there are introns and exons in your DNA and why they get translated into the RNA. You know, it only makes sense not to have them, okay? excuse me, because it does take more energy to copy all of those introns. It's more code, it's more nucleotide, and it's just a lot more work. However, there can be some advantages. It's been theorized or hypothesized, that one possible advantage to having all of these introns is if something happens to your DNA that messes up one of the bases, called a mutation. Okay, when you have something that messes up your DNA, it's not going to hurt the overall organism. Okay, so with all of those introns in there, if the mutation happens in an intron, well, who cares, right? The mutation is just going to get cut out again anyway. If it happens in an exon, then it might destroy whatever protein it is you're making and cause an abnormality. It can hurt whatever the organism is. It can hurt you and me. But again, if it happens in the introns, it's not going to hurt you because it doesn't code for anything. It could also be that for every gene, there can be many different types of mRNA that can be made. Okay, so after you cut out the introns, there's no rule that says you have to put the exons all back together in the same exact order. You can put them together in different orders and it would still make different genes, different proteins. Okay, so again, we don't really know why. Okay, it does take a lot more energy and it just kind of seems silly for our cells to use all that energy in making all of these introns that we're not going to use anyway. But scientists have hypothesized that it's possible. It's due to the fact that mutations in all of those introns is not going to hurt us as much as a mutation in an exon would. Okay, and it also could be that different mRNA can be made by putting the exons back in different orders. Either one could be correct. We don't really know. Why do I keep doing that? Oh no. Sorry. Every 3 nitrogenous bases is called a codon. Each codon codes for 1 amino acid. Okay, each codon is going to tell your cell what amino acid to use. Hopefully you don't hear that weird cat outside. From the last slide, let's try this. Let's try to find UCG. Okay, let's try to. I'm going to be providing you guys on your tests. Okay, so learn how to use this chart. Okay, now there are some special codons that you need to be aware of, that you need to know. Okay, there's a special codon called the start codon, and the sequence is AUG. Okay, it's important because all proteins start with AUG. The protein synthesis, the translation, will not start unless you have this sequence in your mRNA somehow. It codes for an amino acid, called methionine. Okay, methionine. And again, if the mRNA sequence does not have AUG, the ribosome will not start translation. So the ribosome will only start translation if it can find that AUG segment. Once it finds AUG, it can read everything following it and start the translation process to make the protein. Okay, it doesn't matter how many codons are before AUG. You have to have AUG before you start translation. And then the other special codons are called stop codons. Okay, it's similar to the period at the end of the sentence. It's going to stop translation from happening. So you have your start codon, which begins translation, and then you have 3 of them that stop the translation. They don't code for any amino acid. Essentially, what happens is once the ribosome reads the mRNA sequence and it gets to the very end and there's a stop codon, that is just a signal for the RNA to let go of the protein, and the protein just falls off and goes to where it needs to go because the stop codons do not code for any amino acids whatsoever. It just tells them to stop. Now, again, I don't need you to memorize these codons. I just need you to know that there are stop codons and start codons and what their significances are. Significance, whatever. Why they're important. Okay, so in order for translation to occur, you have to have mRNA, right? That's pretty obvious. The mRNA, the messenger RNA, brings the message from the DNA to tell your cell which amino acids to put in what order. mRNA and then attaches to the ribosome. And as the mRNA moves through the ribosome, think of it like I don't know, a scanner. A scanner, like a computer scanner, not a scatter. Okay, so as the mRNA moves through, that ribosome is going to scan the message that's on the mRNA. It's going to read those codons. When it does that, when the ribosome does that, it's going to send a message to the tRNA, which is a separate type of RNA, and it's going to say, hey tRNA, this codon was read. This codon codes for serine. Bring serine. And tRNA will bring serine over. Okay? And as the mRNA continues to go through, each codon is read. The message is again sent to tRNA. tRNA is going to bring the amino acid. Okay. So the three types of RNA. mRNA is a messenger. It brings the information from the gene, from your DNA, to your ribosome. The rRNA is in the ribosome, and it helps to read those codons on mRNA. Then tRNA is the transfer RNA, and it brings amino acids to build the protein. The tRNA is going to provide all of the amino acids for the growing protein chain. Okay. I think this is the last thing I'm going to talk about, which is the transfer RNA. Again, each transfer RNA will carry one amino acid to the ribosome to make protein. It's going to attach by base pairing with it. Well, what does it mean by it? With the mRNA. Okay, so if you guys can, please change the word it to mRNA. It's going to attach the amino acid to the protein chain by base pairing with the mRNA. Okay, because tRNA has what's called an anticodon. The anticodon is the opposite sequence of what the codon reads. Okay, again, complementary is opposite, right? So if the codon reads AUG, for example, the start codon, then on the tRNA it's going to read UAC. Okay, it's complementary to the codon on the mRNA. That's why it's called the anticodon. Okay, and that anticodon is going to be holding on to, okay. So for example, AUG found on methionine is going to have the anticodon UAC on tRNA. Okay, this is your tRNA. The amino acid that is going to be attached to the top. That should actually say mRNA as well. So can you guys change methionine to mRNA? I made a mistake. I'm sorry about that. So here's your tRNA. You have your amino acid attached to the top. It has an anticodon sequence on the bottom. This anticodon sequence is going to try to find the complementary codon inside of your mRNA. When it finds that, it knows, okay, this is the place where this amino acid is supposed to go. I'm going to drop off this amino acid. It's going to polymerize with the rest of the amino acids and form the protein chain. Okay, sorry about that. This has been pretty successful, except for the parts where I messed up, so it's all good. Okay, so again, at the translation, when tRNA brings those amino acids, matches up the codons with the anticodons, and drops off the amino acid, the peptide bonds are going to be formed by the ribosomes to hold those amino acids together and make the protein molecule. When it reaches a stop codon, all the ribosome does is release the protein molecule. Picture is the image of translation occurring. Here is your RNA molecule, or sorry, mRNA molecule on top. This is the message that it got from your DNA, your gene, to make a protein. Okay. The only thing I don't like about this picture is that it doesn't show the start codon for methionine, but that's okay. You guys should know that no matter how many in front there are, it has to have AUG in order for it to start, that start codon. Okay, these are codons. Every 3 letters is a codon. It's going to match up with the anticodon. Notice how they are complementary on the tRNA. Okay, because of that, the tRNA will know exactly where to bring these different amino acids to form the peptide chain. Okay. And that's pretty much it for 12.3. So tonight your homework is to do 12.3, the section assessment, and to print out the homework packet that I have on there for DNA, RNA, and translation transcription. Bring it in tomorrow. Okay, I hope everyone has a great day. Hopefully I was able to finish all of that within the period. Again, your homework, 12.3 section assessment. And please, please, please print out the paper that I have on Edline for you guys. DNA, RNA, translation, transcription. Okay? Cool.